This episode is about a just-published scientific paper about the evolution of hearing, which explores the combined impact of biological and environmental factors on how our ears respond to sound. The study was the brainchild of Dr. Patricia Ballaresque, who got really interested in the different factors, such as our sex or environment, that might have shaped our hearing sensitivity. This groundbreaking study, Sex and Environment Shape Cochlear Sensitivity in Human Populations Worldwide, is published in Scientific Reports. It was led by Dr. Patricia Ballaresque at the Centre for Biodiversity and Environmental Research, CRBE, in Toulouse, supported by the Eco-Anthropology Unit, EA-CNRS, Muséum National d'Histoire Naturelle, at the University of Paris, and the Toulouse Institute of Research in Computer Science, IRIT-CNRS, at the University of Toulouse, and others, including me at the University of Bath and in my previous role at the University of Leicester. So how did the study start? Despite being one of the traditionally recognized senses, the others being taste, sight, touch, and smell, hearing remains an underexplored aspect of human evolution. We know that generally our hearing declines with age, and we know that our modern world is contributing to hearing issues around the world. Recreational noise and ototoxic substances, chemicals such as tobacco smoke, that have an effect on the ear or its nerve supply can cause damage to different parts of the ear. We also know that exposure to chemicals can make ears more sensitive to the harmful effects of noise. And we know that hearing damage from the environment, in the form of noise pollution, is on the rise. This can be the background noise of traffic and everyday activities, such as household appliances like vacuum cleaners or lawnmowers, through to more one-off high noise level events like concerts or sporting events, and so on. So what have been the various factors that have shaped our hearing and our diversity of hearing sensitivities in the human population? I think it's probably a good idea to start with how hearing works. Put most simply, our ear collects sound and then turns that into signals for the brain to interpret. There are three parts to our ear: the outer, the middle, and the inner. The outer part of our ear, which is made up of the pinna, the bit of the ear that you can see, and the ear canal, collects sounds from our environment and channel them down the ear canal to the middle ear. Here, they cause the eardrum to vibrate. On the other side of the eardrum are three tiny bones, called by the Latin names malleus, the incus, and the stapes. These translate to hammer, anvil, and stirrup. They're lined up in a configuration with the malleus next to the eardrum picking up its vibrations, the incus in the middle, and the last one, the stapes, transmitting the vibrations to the inner ear. The inner ear is made up of the vestibule and the cochlea. The cochlea looks a bit like a snail shell. The sound vibrations entering the opening of the cochlea travel down it, and the cochlea turns this information into nerve signals, which then travel to the brain to be interpreted. Currently, your brain will be interpreting you listening to this podcast. There's two main measurements of sound: frequency and amplitude. Another name for frequency is pitch. Drums have a low sound pitch, where a whistle is high-pitched. Frequency is measured using a unit known as a hertz, and human hearing range is between 20 hertz and 20,000 hertz. Anything above 20,000 hertz is known as ultrasound. Amplitude is a term for loudness, and that is measured in decibels. Normal conversation tends to be at about 60 to 65 decibels, where vacuum cleaners are around 75 decibels. In terms of the evolution of communication and hearing, the thinking around animals is the acoustic adaptation hypothesis. It predicts that the sound properties of communication would have been selected for the optimal transmission for the habitats that the animals are living in. For example, animals living in habitats like jungles are going to make and hear different sounds than those in grasslands versus those who live in the ocean. So that got Pat thinking. It would be interesting to look at factors that affect human hearing. The cochlea, as we know, plays a central role in hearing, converting sound waves into neural signals. You can measure cochlear sensitivity using something known as transient evoked otoacoustic emissions, TEOAE techniques, which measure the ear's reaction or sensitivity when it receives sound. More specifically, the cochlea's ability to produce and transmit an acoustic response after being sound stimulated. It's often used by ear, nose, and throat specialists as one measure of hearing sensitivity, often to check hearing in newborns. We already know that the sensitivity of the cochlea varies across individuals due to biological factors such as age, which ear is it, left or right, and sex. For example, we know that hearing declines with age. We know that the right ear is typically more sensitive than the left, and women seem to have higher sensitivity than men, particularly above 2000 Hz, at least in European populations, since other populations have not been investigated to the same degree. We know that ethnicity can potentially also play a factor, though this too is not well studied, but suggests that there may be evolutionary adaptation to local environments. But so far, the influence of these on hearing sensitivity in humans has been underexplored. This study aimed to look at cochlear sensitivity through TEOEA measurements across global human populations and evaluate the relative impact of both endogenous factors such as age, sex, and ear side, as well as exogenous factors such as language, environment, and ethnicity on hearing variability. We analyzed the hearing of 450 individuals across 13 populations in Ecuador, England, Gabon, South Africa, and Uzbekistan. These populations were selected to capture a wide range of ecological and cultural contexts, including underrepresented rural and non-European groups. We were interested in whether people were living in urban environments, rural forest, plains, steppes, or at altitude. We categorized participants into groups based on their age and sex, language family, ethnicity, and the environment they were living in. So what did we find? Sex was found to be the leading factor explaining cochlear sensitivity differences, with an average of 2 decibel difference between men and women across all populations. This biological factor best accounts for variation between individuals. Following this, the ecological environment in which individuals live best explains the remaining differences, which can reach an average of 3.5 decibels between landscapes. We saw significant differences between populations in a forest environment and populations at high altitude, with the forest populations having higher sensitivities than those at high altitude who had the lowest sensitivities. Of the endogenous factors, sex emerges as the most influential factor, with women consistently showing higher sensitivity across the entire frequency spectrum tested and across all populations. Several hypotheses can help explain these findings. First, sex differences may be partly attributable to varying levels of hormone exposure during development in the womb. There's also structural differences in cochlear anatomy between men and women, although these don't fully account for the differences seen. In addition to heightened cochlear sensitivity, women also perform better in other hearing tests and speech perception, indicating superior function in both the peripheral auditory system, the ear, and the central auditory pathways, which is the nerve signals and the brain interpreting the sounds. Women show a higher prevalence of hyperacusis, which is a disorder in loudness perception. Given the well-documented detrimental effects of noise on overall health, such as reduced sleep quality and increased cardiovascular disease risk, maintaining heightened cochlear sensitivity in noisy environments has potential implications for understanding sensory processing and health in diverse environments. While a decline in cochlear sensitivity with age is seen, its influence is not as strong as sex on hearing sensitivity, and ear side is the least influential factor in explaining hearing sensitivity, though it does reaffirm a subtle right ear advantage. This is likely due to the right ear's primary role in auditory processing within the left hemisphere of the brain, which is crucial for human abilities such as speech perception and production. Our results offer pioneering insights into the role of non-biological, so exogenous factors in shaping cochlear sensitivities. We found that the environment in particular not only affects the response to volume, but also to the range of frequencies of sound perceived. In fact, population, environment, and language all significantly contribute to the variation in hearing across human groups. We aren't quite sure why. It could be that the entire body is affected by the environment, elevation, and temperature, and that then affects the cochlea. Or maybe it's long-term adaptations to varying soundscapes. Or maybe it's different levels of noise exposure and exposure to chemical compounds that are toxic to the ear in some way. The greatest contrast was found between populations living in sheltered forest environments and high-altitude Andean populations in Ecuador. The forest environment, characterized by a rich biophony, sounds generated by non-humans, and minimal sounds generated by humans, suggests that higher cochlear sensitivity in these populations may reflect a number of things. It could be an innate sensitivity to non-human sound environments, or an inherited trait favoring heightened sensitivity in biophonic environments where vigilance is essential for survival. It could also be being reinforced by the absence of noise pollution or ototoxic substances such as chemicals. Those populations at high altitudes had lower response levels. This reduced sensitivity could be due to a number of factors. The effect of atmospheric pressure on TEOAE measures, potential sound reduction in high-altitude environments, which might lower the need for high auditory acuity, or physiological adaptations to hypoxia. We do know that high altitude causes low blood oxygen levels, which affects the body's physiology, and that may include damage to the cochlea. There's also a difference between urban and rural populations, with a shift towards higher frequencies in urban populations, which may be a response to the low-frequency traffic noise prevalent in urban populations. We know there is also a similar shift in bird vocalizations in areas where there are more humans. These results highlight the diversity of mechanisms through which we perceive and process sound. The differences between men and women could be explained by anatomical, hormonal, or genetic differences. Differences according to ecological landscapes, on the other hand, could result from two pressures. Either ancient adaptations from human colonization of these environments long ago, for example, altitude, or that there is plasticity in our hearing sensitivity, such that it can change in response to an environment that has changed more recently, such as urban versus rural. While age is a known factor in hearing decline, this study shows that this factor is overshadowed by the effects of sex and the environment. This study highlights the crucial influence of environmental factors on cochlear sensitivity and underscores the need for further research to determine why. We know that we humans are continuing to evolve, so the question here is, are these differences just our ability for hearing to change in response more generally to different environments, or are there new genetic adaptations that are involved? Watch this space.